Hi, so how is everybody doing today? Can you hear me? Hello? No, this wasn't even the one I dropped. Let's try this one. How about this one? Can you hear me now? Yeah, hopefully. It is blinking, so maybe I'll just speak on this one here. How about this one? Can you hear me now? Hello? Hello? Okay, everybody, so it seems like the mics are not working very well. Hello? All right, so we got two bad mics here, and this one here, I am not sure if it is working or not. Well, it's not green here. Maybe it's not connected. Okay, everybody, so let's focus here. We gotta cover a lot of replication. Everybody here? Yeah, so? I mean, half here, half there? Or can you hear me okay? Yeah, everybody awake? Yeah. So when is your next test? On the 10th, right? October 10th, right? And is it in person? Yes, everybody gotta be here. Yeah, and no cheating, right? What do I have to say about that? Oh yeah, I was asked if you get here a little bit after 2 o'clock, if you can still come in the class and take the test. My answer is yes, as long as you're very quiet and you do not disrupt, the class. So, if you're stuck in traffic, which happens very often around this, part of South Florida, or, if you're rushing to get from one other class to this class, don't go crazy trying to run a marathon to get here. You get here, you get in, you take your test, and I don't think this class is used afterwards, is it? Do you know if there is another class after us? No? All right, so if you need a few more minutes to finish up your test, it's okay with me. I don't like to put a lot of pressure because the nature of the subject is already difficult to digest. So the least amount of pressure I put on you guys, the better I feel. And hopefully the better you feel as well. All right, so did everybody notice all of those Jove videos that I have put up? Yeah, and I'm sorry that it makes Canvas to look a little bit crowded, but that's the best I can do at this point. It's a lot of learning curve to use these resources. But I find those videos to be useful. And have you seen the practice quizzes? Yes. Have you seen the latest one I put over this weekend? It has, I don't know, 100 something questions. No? No, make sure you look at it, because, think about it. If we have 70 slides in a class, and I put 100 and something questions in there are only so many questions I can ask about the subject, right? So if you understand those questions, yeah, come on guys, let me not be screaming over the chat. Okay, so if you know those questions, you understand those questions, most likely you're going to find something similar in the test, okay? If you don't understand what's going on with those questions, what the question is asking, what the thing is talking about, get your study groups, okay? I would advise at the most 5 people per study group, otherwise it's a party, okay? And make sure that you focus on the subject. Don't start talking about everything else that goes on, because that is very interesting during this time, right? Just for curiosity, how many people of you watched the Jimmy Kimmel show last night? No. Okay, I did. Oh, but he's back last night. He was back. Yeah. Okay. Anyway, we're not getting into politics, but there was one thing that I want to bring up. Have you heard the latest about the president's say on not using Thailand? Okay, before you judge, there is a bit of truth about that. Yeah, it's scary. When we get to nucleotide metabolism, which I'm not going to drill you too much with it, when we get to that, and antioxidant function, we'll bring up the Tylenol issue a bit more. So this is going to be for scenes of the next episode. Over here, In this cell, let's say that is not very much expressed. So it gets close to the periphery of the nucleus. But in another T cell type that has other types of gene expression, then that chromosome might actually get closer to the center. Okay, so they like to stay in their own places. Now, parts of the chromosome that get expressed, they are close to the center, and then you have a lot of tunnels and branches that the pieces of chromosomes can shift from one side to another, get expressed, and then go back and get regulated. Okay. So chromosome territories, the position of the chromosomes really have to do with the cell type and how the cell got differentiated, terminally differentiated. So close to the edge, they tend to be quiet. So here we go for levels of packing. It's also called compaction. Okay. So first thing you're going to have is, of course, your DNA double helix. Then it starts to get folded around those proteins that we have to know a lot about them, right? Around those histones. And once you get around the histones, you get these beads on the string type of setting. And this gets to fold the double helix on itself, and you go from a very small 2 nanometer size to a larger, over 10 nanometer in size. When you look at those folding, those wrapping around your histone cores. Then if you start packing those histone cores, really the nucleosomes, you start packing them close to each other, then you get this fiber that is called the chromatin fiber, and that is about 30 nanometers wide. Okay, so this is when you say chromatin fiber, you say that's a 30 nanometer fiber. Then you get this chromatin and start wrapping around itself, and then you get the bigger type of fiber, and with that you're gonna get, you know, your chromosome. Okay. Remember that each arm is a chromatid, so these two are sister chromatids. And you do have the replication of the sister chromatids during your S phase of mitosis, so during the synthesis of DNA of mitosis. Okay. So this video I really like that it shows you the DNA packing. And all right, so this thing is kind of low, but you should be able just to see it. So what is this guy up there? Those are histones, associating histones, right? So you get your nucleosome here. You see the chromosome packing around the histones, and then you get those nucleosomes, and then here you get your 30 nanometer fiber, and they keep on packing. And wind around the fiber, wind around itself, and then you get your chromatid. All right, so that's a nice microscopy image. Well, she says not really present. They are present, but you cannot really quite see them unless they are condensed. So this you see the cell dividing and the sister chromatids being pulled apart. Go, cell division, nice, beautifully done. Okay, so here comes your first question. So we do have some cool points today, and you should definitely know the answer for this. Oh my goodness, this thing is going quite quickly. I'm going to stop at 30 seconds. Make sure you get your points. Okay, 5 seconds. Anybody need more time? No? All right. What is your answer? A. 2 sister chromatids. Okay. All right. So here, nucleosomes. What are the nucleosomes? You have the DNA winding around twice, about twice. Okay, look at this, twice. Okay, around those proteins. So you have DNA associated with proteins. Now look at this thing over here. You have each one of those proteins called H. H is for histone. Okay, I got a little mnemonics on how you're going to remember the name of those proteins. All right, so histones over here. Look, you have the tail of the histones that are coming out from that association. So you have tails sticking out everywhere, okay, out of those histones. So how is it that you get this histone core? You are going to have packing of 8 proteins. So if you open this, you're going to have 4 proteins and 4 proteins. They're going to associate and make this little core. Okay. What are the proteins that are associated with the core? You have H2, H3, and H4. 2 flavors of H2. You have H2A, H2B, H3, and H4. That forms your core. You don't have H1 at the core. Okay. So those guys are going to make the core. DNA is going to wrap around it. So here you have DNA wrapping around that. Are histones the only proteins associated with DNA? No, you have other non-histone proteins that also associate with the nucleosomes and stabilize the structure. Okay, but you don't have to worry about those non-histone proteins. So what do you need to worry about? You need to worry about the histone core. And then you're going to have this other histone that is going to be H1 that is not associated with the core. It actually likes to associate with the naked part of the DNA. Okay, so here, what happens here? This histone, you're going to have only one copy of histone that is going to associate with this nucleosome. So look what this histone is doing. It is associating with the part of the DNA that is not getting protected by those histone proteins. So we call this part of the DNA right here, we call it naked DNA. Okay? It is naked. Is it a good thing for the DNA to be naked? No, because it can be attacked by endonucleases, right? Enzymes can go there and recognize some sequence and chop up the DNA. So not a good thing for the thing to be naked. So this guy over here goes over there and associates with this linker. Okay, besides being naked, you also call it linker DNA. Okay, and let me see here. Yeah, histone, the H1 also helps to stabilize the chromatin fiber. And another question. What is the primary responsibility of histones? You didn't even start talking about damage, right? So there's only one obvious answer. Okay, 5 seconds. Hey, what is your answer? Huh? C. Yeah. Okay, so for the next question, I'll put on the confidence level. Okay, so here, this is important things you have to know about histones. Okay. Oh, how are you going to remember that H1 is your odd histone? How do you know the name of histone in the first place? Well, when you look at the structure, you look at your structure of those nucleosomes, and you get 2 of one type and 2 of the other type, okay? So what do you see there? You see 2 little balls of each type, and you say, oh, it looks, okay, everybody is over 18 over here, right? Can I tell you a bit of a dirty joke? Yeah. Yeah. It looks like his stones, kind of. So, all right, 2 balls are his stones, Okay. What about H1? H1 does not look like two balls, it looks like a little stick. So you get only one stick and two balls of each type. All right, so now even if you want to forget about your histones, you can't. So that was my little, fancy joke. So what can happen to histones? A lot of things can happen to histones, okay? First thing that can happen, you can get them inactivated. I'm not talking about kicking. I'm talking about other forms of inactivating these stones. Okay, so you can put a little bit of inactivation. You can put a lot of inactivation. Okay, little bit of inactivation means you can put a non-polar group. There is a methyl group over there, and look, methyl group is covalently bound to this positively charged residue of amino acid. What is that amino acid? The name is right there, it's lysine. Okay, it's positively charged. A lot of positive charge to the histones because they want to associate with the negatively charged phosphate groups on the DNA. Okay, so now you can put one methyl group over there and you can inactivate it a little bit more. You can put two methyl groups or even three methyl groups, and you get this cloud of stuff here that is really stabilizing that positive charge on lysine. If you really stabilize the positive charge, it means you make this whole part of this molecule basically non-reactive. And the charge is over there, it means that it will interact with the negative charge of the DNA, and you're not going to be able to pluck it out. Okay, so adding methyl groups to lysine is going to make that piece of DNA inactive, meaning not transcribed. Meaning if it is a gene, it's going to be silenced. So adding methyl groups to histones is going to get your gene quiet, silencing gene expression. Okay. On the other hand, if you put another group, like an acetyl group, to this amino group over there, what is going to happen? You're going to neutralize your positive charge. That means, why? Because this thing is negative charge, that thing is positive charge, you neutralize it. That means with a neutral charge, this thing is not going to be interacting with the negative charge DNA as well. Okay, so you can pluck out the interaction of histones and you can expose that part of the DNA, meaning that if you have a gene that you want to express, you can actually get to the DNA and you can get to express the gene. So acetylation means you're going to be expressing genes. You're going to be reaching the DNA, you're going to be expressing genes, right? So methylation is silencing, acetylation is gene expression. Okay. Now, let me see here. Besides the lysine, you have other amino acids like serine that can get a group, a functional group, meaning a phosphate. Remember the three amino acids that you can get phosphates on? Serine, threonine, and tryptophan. Tyrosine. What? Forget tryptophan. No, I was thinking about tyrosine. Tyrosine, not tryptophan. That is too non-polar. Tyrosine, serine, and threonine. All right, so this guy is serine right there. It has OH group. This OH group can serve as landing site phosphate. What charge is phosphate? Negative charge. Is it gonna like to interact with DNA? No, it's gonna be repelling DNA. So this is another way to get histones away from that DNA interaction. Okay. So now, histones cores, H2 and H3 and H4, okay, they can get these modifications, right? The enzymes that do the modification, you call those the writers. Okay, so what kind of enzymes? They can put methyl groups, they can put acetyl groups, or they can put phosphates. We just looked at those. What are the name of the enzymes? Methyltransferases, acetylases, and kinases. Those are the writers. They put stuff on the histone. On the other hand, you have the enzymes that take away the stuff from the histone. So those are obviously the erasers. What are their names? The methylase, the acetylase, and phosphatase, not kinase, not the kinase. Okay, so kinase puts in, phosphatase takes it out. All right. So you got your erasers, you got your writers. Okay. So what are the modifications? You got here acetylation, methylation, and phosphorylation. Where is that happening? At the amino terminal tail of your histones, and specifically in lysine residues. Okay. All right. So those modifications of the histones, they are actually writing a type of genetic code that has nothing to do with the sequence of nucleotides that you find on the DNA. Okay. So this is actually really simple. Genetic code, when you think about genetic code, you think about A, C, T, G stuff on the DNA. But this is not related to nucleotide coding. This is related to epigenetic modification. So this is a type of epigenetic code. Okay. So imagine all the cells of a particular type, that those cells are differentiated. They are going to, when the cell divides, they are going to carry this type of histone coding to the daughter cells. So this is a type of epigenetic modification that gets carried on to the daughter cells. Okay? So it's important for keeping the type of cell in with the specific gene expressions that the cell type has to have. Besides histone modification, you also can have some chromatin modification that we're going to see also next. It has to do with this histone pushing around. Okay. So histone code has to do with epigenetics. Okay. Besides modification on the histone itself, you also have certain histone variation or alleles of histones that are important for the histone code as well. But we're not going to get into that. It's too much detail. Okay. So depending on how those histones get packaged, you're going to have certain gene expression. So we know about the histone code. Now, another tool for epigenetic modification, it has to do with this chromatin remodeling business. Okay, so sometimes you want to access certain genes that are packed with these histones, so you need to move the histones around, either by modifying them or by physically pushing them around. So there is this protein, or this complex of proteins, that are called chromatin remodeling complex. Thank goodness the name is simple and easy to follow, right? Now, these chromatin remodeling complexes, okay, they get over here to the nucleosome, and they are going to push this thing around. But in order for this thing to be pushed around, you need energy. Okay, where is the energy coming from? It's coming from ATP hydrolysis. Okay, so this thing is ATP dependent protein complex that moves these nucleosomes around. So ATP dependent chromatin remodeling complex. So the part of the DNA that gets exposed is the part that's going to be expressed. Okay. Okay, so when you look at the chromatid, you're going to see that some parts are not as packed as other parts. So the part that is not as packed, you call that the euchromatin. The part that is really, really packed, you call that heterochromatin. So these different shading patterns are really cell-dependent. Okay. So you have chromatin structure. Okay. So all these things are really making sure that your epigenetic code gets transferred to daughter cells, and how you're going to assess genes. All right. So chromatin variation, different types of chromatin. Remember here you have chromosome. Here is also a chromosome, right? This guy is during synthesis. This guy is during not synthesis. I forgot the name of that. How do you call the place, the time that is not synthesis? It's not senescence. Interphase. All right. So interphase or synthesis. Now, if you look at this thing here, you see that you have the centromere right in the middle. When it's right in the middle, you call it in the middle, so it's metacentric. If it is not in the middle, it's pushed either to this side or a bit down from the middle, you call that sub-metrocentric. Well, this thing looks like on top, but yeah, call it sub, okay? If it is all the way at the tip, close to the telomere, so you call that telomeric, right? Telocentric. And let me see here. Oh, you also have this variation of submetacentric that is closer to the telomere, but not all the way close to the telomere. I get concerned that you really know these three types over here, okay? Because this is a bit confusing with that type. So these guys here are, if you look at the chromosome structure, you should say which one it is. So euchromatin, your fibers are less condensed. Heterochromatin, you have highly condensed fibers. Where are you going to find heterochromatin? You're going to find it close to the centromeres, at the centromeres, and also at the telomeres. You're going to find heterochromatin. Euchromatin, all in the space that is not in those spaces. Okay. So here, more on chromosome structure. So a lot of these features of the chromatin, those are inherited. So that is all part of your epigenome, right? So we call it epigenetic inheritance. Okay, so you can read that on your own. All right. Important things that we should know about this epigenetic inheritance is that we don't see that in prokaryotic DNA, because prokaryotic DNA is quite simple. You don't have those nucleosome structures. You don't have histones in prokaryotes, right? Because you have the little circular DNA that is already stable on its own, and usually the bacteria has to make use of whatever is available in that small circular DNA. All right. So here a bit more on those epigenetics. Those are alterations that affect the expression of genes, and they do not have to do with nucleotide code, okay? You inherited them, and usually they are fixed for a particular type of cell. All right, so here a bit more on epigenetics. You can read that on your own. Okay. All right, epigenetics, cool things about epigenetics. That's the part of your genetic makeup that you can actually modify. Okay, so the epigenetics, it's not really fixed in stone. Yeah, no pun intended. So it's not a fixed thing. You can modify your epigenetics. Right? Actually, there are some cell types that tend to modify the epigenetics and become something that they're not supposed to. So you see epigenetic modification in several or many types of cancers. Okay, so modifying those histones and gene expression that happens to cancer cells. Okay. Now you also see erasing epigenetics completely when you have formation. Yeah. You modify epigenetics. You can modify epigenetics to a certain extent. Because when you talk about epigenetics, you put like a lot of things in one bucket. When you say epigenetics, it's everything that is not DNA code. Okay? So epigenetics of differentiated cells, those tend to be permanent because they have to go from cell division from mother cell to daughter cell. Other types of epigenetics can be changed. Because when you look at patterns of gene expression, and you look at certain cell types, for example, central nervous system cells. So when you look at your neurons, they have certain pattern of gene expression that has a lot to do with epigenetic modification. Now look at this. Many psychiatric disorders have to do with strange type of epigenetics, but those can be modified. And it's really, really interesting when you stop to think about one simple example of epigenetic modification that got modified, and I don't really know how. There is a particular anti-depression drug that is also used for people who are trying to quit smoking, right? And that drug allows you to not act very quickly on impulses, okay? So let's say it's good, it's useful for treating depression, and it's also useful for giving you a bit more of a willpower when you reach for the pack of cigarettes in case you're a smoker and you have this kind of like, drive, like I need a cigarette. I've seen people who couldn't really get rid of smoking ever. So you give this drug and you can control your impulses a bit more. But there is something else this drug does. It stops people from biting their nails. Like, do you know biting your nails has to do with genetic expression of something in your neurons? I had no idea. How did I notice that? Well, because I was taking the drug and I noticed that all of a sudden I had nails and I wasn't interested in biting my nails anymore. I'm like, oh, that is odd. So I started to look it up. It's like, look at this, it's a side effect. So there was some kind of epigenetic modification of some of my neurons that were telling me to bite my nails. And they got changed. I was not interested in biting my nails anymore afterwards. I was like, this is so odd. I mean, this is a drug that is affecting neural expression. Yeah. Yeah, Wellbutrin. You got it. Yeah, that's the name of the drug, Wellbutrin. Okay? And you have also genetic form. Okay, so, all right, so this is actually scary. Okay? Because think about what else can modify your neuronal function? As I mentioned in the first class, you got your microbiome, right? You got those little bugs that are sending messages to your central nervous system and telling you how to think in a certain way. Can you modify those messages? Yes, you can. Yeah. Can you get rid of the bad bugs? Oh, yeah, you can. Is it difficult? Yes, it is. It's a lot easier to get rid of the good bugs. Don't ask me why. It's always like that. So, but yeah, psychiatric disorders, they have a lot to do with epigenetics. Besides drugs, what else can influence your epigenetic expression? Yeah. Yeah, but those can be considered as bad drugs. But yeah, chemical exposures can do that. Yeah, UV light can definitely mess things up a lot. But it can also be good, give you vitamin D. Yeah. Your diet, absolutely, yeah, diet can modify your microbiome, can modify. But I was thinking about something that usually people don't think much about. Nah. It depends on how toxic they are. So. Anybody meditates? Yeah. Meditation can modify your epigenome. And that is really, really cool. Right? Because you can treat several psychiatric disorders with meditation. I mean, really? What do you do when you meditate? Usually you're trying not to do anything, you're trying not to think about anything. But at the moment you sit down and you start to think that you cannot think about anything, that's when you start thinking about everything. It's kind of difficult, right? But if you can, even guided meditation, if you can, sit down for 10 minutes a day and meditate, guided meditation, that is going to enhance, guess which part of the brain gets enhanced by meditation? Memory, recall, and retention. It goes well before a test, right? Anyway, we get to the more epigenetics when we get. But yeah, that's a good thing. You can modify your capacity to remember things by meditating. Take 10 minutes out of your busy day, sit down and meditate, and you're going to do better in your tests. It's not a secret, right? But I want to make sure that I tell you that. Okay. Oh, healthy aging. Have you seen some people that they seem that they never age, even without plastic surgery? They live a cool life. They meditate, and they don't stress out about things, about many things. All right. Healthy aging. Okay. All right. So that's a lot about meditation, about epigenetics. So here comes another question for you. Chromatin remodeling complex are essential for what? Rule of thumb is that if I did not talk about something, it's probably the wrong answer. All right. So anybody need more time? Okay, what's your answer? B. B. All right, so finally, we're going to get to replication. Everything you need to know about replication out of this class, right? So let's listen up here. Okay, you can read that on your own. Let's get to the stuff that is really important. All right. What do you need when you do DNA replication? You need to make sure that the thing is reliable. So you need to have high fidelity, okay? You have to make this thing work every single time. So high fidelity, very, very important for our replication. And what is going to happen with DNA replication? All DNA replication is going to happen in this semi-conservative fashion. That means your starting DNA, so here's your double helix. When you do the replication, you're going to end up with 2 of those double helix. One of those new double helix is going to have the old DNA, and the other strand is going to be newly synthesized DNA. So this is what we say, this is what is meant by semi-conservative type of replication. Okay? We discovered that, well, there was these guys, Mendelssohn and Stahl. Strange. Oh, great, now I don't remember if I have this slides here or there. What did they do? They made heavy DNA using radioisotopes. So, I mean, they use phosphates that were heavier than regular phosphate. So they made heavier isotopes, and they were working with bacterial DNA. Okay, so they had here a flask that they were growing bacteria, and in this bacteria they were adding heavy isotopes. So everything here in the DNA was heavy. Okay. Then what did they do? They allowed this thing to undergo replication, one round of replication, and the nucleotides that they added to here, the nucleotides were regular weight, was lighter. Okay. So now they get nucleotides that are lighter, and they're going to end up with something that has old DNA and new DNA. So the blue one is heavy DNA, the red one is light DNA. Then what did they do? They spin this thing down, they put it in a tube, and they spin this thing down, and they look where in a gradient of diffusion, in a density gradient, where is it that they're going to find the DNA? If the DNA is all heavy, it's going to go to the bottom because it's heavier. When you centrifuge, things go to the bottom. If the DNA is light, it's going to be at the top. If the DNA is mixed, it's going to be mixed right in the middle. Okay? You see what I'm saying here? Okay? So with that experiment, they figured out that replication of DNA is semi-conservative. Because after the first round of replication, they got all the DNA to show up in only one band. Okay? They did a bit more of experiment adjusting to make sure that it wasn't dispersive, but you don't have to go into that many details. Okay? So here, talking a bit more about the types of DNA that are important for inheritance, right? Remember here in your eukaryotic DNA, you have also beautiful linear DNAs, right? When you look at prokaryotes, you're going to have circular DNA, which is right there. So the circular DNA is not the only type of DNA that the prokaryotes have, not genomic circular DNA. You also have smaller little circular DNAs in prokaryotes that you're going to find dispersed throughout the cytoplasm. Okay? So the smaller DNA, those are called plasmids. Okay? So genomic DNA is the bigger one, also circular. Smaller ones, those are the plasmids. Okay? Now, interesting thing about the plasmids is that the genes that are encoded by the plasmids, they can be shared between different bacteria. So this green guy over here can get together with another bacteria that does not have that little plasmid, and it can replicate the plasmid independently of the genomic DNA. You can replicate this plasmid over here, so let's put another plasmid over here, and then you donate that plasmid horizontally to another bacteria that is close by. And this is how bacteria acquire gene expression that they didn't have before, especially resistance to certain antibiotics. Okay? So this piece of DNA can be shared. So now, if this new bacteria here has the little plasmid that expresses a gene that makes it resistant to certain antibiotic, so this guy here is also going to be resistant to the antibiotic. Okay? So we have seen this before and that before, so now we see plasmids. And they replicate the same way that the other DNA replicates, semi-conservatively. But the one thing that is cool about plasmids is that they can replicate without cell division. Okay? If you look at the genomic DNA, that one is only going to replicate when the bacteria is ready to divide. But this guy here, they can replicate on their own. Does not depend on cell division. All right, so now looking at how is it that replication is happening. Replication is going to start at one specific place in the DNA that has a certain sequence of DNA. So there's some C, T, A, T, C, C, something that is a piece of the DNA, and that is called, the segment of DNA is called the origin of replication. When you look in bacteria, bacteria is going to have only one origin of replication. Okay? So origin of replication is going to start there. And then replication is going to proceed, remember, 5' to 3', because the 3' has the OH group, right? 5' to 3'. All nucleotide synthesis happens 5' to 3'. Not only DNA. All RNA synthesis happen 5' to 3'. This is a rule, okay? All right, so how is this going to happen? We're going to see, we're going to have this replication bubble, and you're going to call this whole thing theta replication, and it's going to work bidirectionally, so the bubble is going to open on the two sides, and you're going to have replication forks on both of the sides. All right, so here is your bubble. This is your circular DNA for bacteria, okay? So first, you're going to have to open up the DNA and to allow proteins to get in there to start the replication, okay? And then you're going to have this replication that happens bidirectionally. And then you're going to have this new strand that is made, and then they're going to separate, and you're going to get your fully replicated DNA. When you look at eukaryotic DNA, now you have a problem, because bacterial DNA is actually small, and processivity of the DNA replication machinery, right, DNA polymerases, it's good for bacterial replication. But when you look at how huge your eukaryotic DNA is, then there will be a problem if you have only one original replication, because you'll never be able to replicate the entire chromosome in fast enough time for maintenance of life. So to replicate all the DNA that you have in one cell, it would take about, I don't know, a month. So it would not be feasible. It will not be compatible with life. So in order to maintain life, linear DNA will have many, many origins of replication. Okay, so this is the first difference between your prokaryote and your eukaryote DNA replication. Prokaryote has only one ORC, original replication, and linear DNA has many, many, many origins of replication. This is the time when I do, I usually do a little class exercise of the wave. Okay, we can actually do it here. So if you go like to this part here, if you think about prokaryotic DNA replication, okay? So if you start like on this side here, and then over here. Okay, so first we go on this side. Everybody knows the way? So very, very, you start the way, and then the way from here. Okay? You have the way. Some people are beginning before everybody else. That's when you're going to have a problem. We want our interpretation. I don't want to give me a bit lonely. Let's make it go away. That's the interpretation. Now, eukaryotic DNA has many amino acids. So everybody here and everybody is going to get at the same time. Ready? Okay. 1, 2, 3, go. In the middle. Okay, so like this way, go this way, this way, this way, you know what I mean? Okay, this way again. 1, 2, 3, go. Okay, good. And then we took half the time. So let's let's wake up with a wave kind of exercise. All right. Eukaryotic DNA, many, many, many origins of replication. Okay? Okay, so what do you need to replicate? Anybody run PCR before? Anybody been in a lab run PCR? All right, so you know everything about what you need about replication. Because if you forget any of those ingredients, your PCR doesn't work, right? So let's see here, what are your ingredients to replicate DNA? Same thing you do in the lab. What you're gonna need? First thing, you're gonna need your template. You're gonna need a thing that you actually have to replicate. Okay. Then you're gonna need your raw material. You're gonna need your DNTPs. You're gonna need a nucleotide, 3 phosphates. D, because remember, it's deoxyribonucleotide. Okay? You know all of this, right? So deoxyribonucleotides, and you're gonna need your enzymes, polymerases. Okay? So you're gonna need polymerase. And you're also going to need a ligase. Very important if you're cloning stuff. Okay? Ligase is really, really cool. All right. So this is what you need, basic need of replicating your DNA. Enzymes, okay, enzymes is polymerase and ligase. Okay? So those are your raw materials. Okay. So remember, you are going always to synthesize 5' to 3', and that's because of your leading group right there. We mentioned that before. Okay, so direction of replication, okay, you can read that on your own. And here we're going to have your 2 strands that are getting replicated. They are going to have to do the replication at the same time. But now you run into a problem with your lagging strand. Well, let's see first what happens to the leading strand. You start 5' to 3', and you have continuous replication. The fork is opening this way, so the replication of this other strand has to go backwards, and your enzymes cannot do the opposite, 3 to 5. It cannot do replication. So this strand also has to be replicated on 5' to 3', but you have to do a little trick in order for this thing to get replicated. Okay? So this is when you have your continuous replication on your leading strand and your discontinuous replication on your lagging strand. Well, the term lagging is actually a bit misleading because lagging, it means like it's lagging behind, but it's not really. The replication on both strands are happening at about the same speed. Okay, so whatever is happening on the lagging strand has to be happening really quickly to be catching up with whatever is happening on the leading strand. Okay. So the the fragmented type of replication, that's on here your lagging strand, okay? And the pieces that get replicated at one time, you call those the Okazaki fragments, okay? Because Okazaki was the guy who discovered this type of replication, this continuous type of replication. Besides the theta replication, you can also have another type of replication in prokaryotes that is called rolling circle. Okay, we're not really going to study a lot of the rolling circle. It is simple, but it's just a different way, different mechanisms. You do not have Okazaki fragments on a rolling circle because you only do one template, is only a leading strand, and that's it. So we are focusing here on theta replication that is very similar to eukaryotic replication. Okay. So processes of DNA replication. What is going to happen here? First thing that you have to do, you have to start it. So that's initiation. Then you have to pry the DNA helices apart, right, to replicate semi-conservatively. So you have to unwind the DNA. Then you have to do the replication and elongate on both strands, and then you have to finish it up. So it totally makes sense. Okay. So to start, you are going to start at the ORIC, origin of replication. Okay. Who is going to start? In prokaryotes, you're going to have your initiator protein that you call that thing DNA-A. DNA-A. It's not DNA, it's DNA-A. Okay? Okay, so that's a protein. Okay, it's called DNA, but it's a protein. That's your starting protein. Then you're going to see step by step, okay, what's going to happen here. Remember, you have a single, oops, it's my husband, he knows I'm teaching. Yep, it happens all the time. Can't talk right now, honey. All right, so what's going to happen here? We are starting it. What is the first protein that is going to interact with your DNA? You call that DNA-A. All right, A gets there. What is DNA-A going to do? It's going to associate with this double helix. Okay, what is that part of the DNA that this thing is associating with? Is this green segment right there. What do you call the green segment? Origin of replication. That's only this part that can associate with DNA-A. No other piece of your DNA can interact with those proteins, only this piece. Okay? So DNA-A gets there and starts to get this little coil, this little loop in the DNA. Okay, so it bends the DNA a little bit. After that, you are going to have unwinding of the DNA. That means you're going to have a helicase that goes in there and starts to pry those two helices, just two strands apart. Okay? When your strands are apart from each other, so this is apart, this is together, this is apart. Okay? So now this piece here that is apart from each other, because of helicase activity, what could happen here is that they could just snap back together. And that would be totally counterproductive. So in order to avoid those things to snapping back together, you bring another protein that is this single strand binding protein. So now single strand binding protein, guess what's going to do? It's going to bind to the single strand. Yay. All right. Bind, 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 bind. When your single strand binding protein binds to the single strand, that looks like a mouth with teeth. But anyway, it's not going to allow these strands to interact and snap back together. Keeps them apart. Now, this single strand binding protein, they're really, really interesting because they are going to avoid the DNA interaction, but at the same time, they are going to keep a little bit of that nitrogenous base exposed because your polymerase has to recognize that base and it has to do the polymerization. Okay? So this is a really cool type of protein that avoids rewinding and also allows polymerase to work. So first thing you did, you pry them apart, use helicase, then use single strand binding protein. Now, if you look at the piece of DNA that is at the edge here of opening up the loop, this part is getting tangled up. It's getting coiled and coiled and coiled. If you don't release the coil, you're going to break the DNA. So this is when you get this DNA gyrase to go over here and release the tension. Okay? So several things going on right here. You open up the loop, you stabilize the loop, you relieve the tension. So here you go for your unwinding, helicase right there, then you go your single strand binding protein. All right. Now, these helicases, most of the helicases that are doing the relief of the unwinding here, they actually use ATP to do the unwinding. So this helicase is a type of topoisomerase that uses ATP to do the unwinding. So we mentioned that. So here's a nice picture showing the bidirectionality of this. Okay, oh, and you have another enzyme here that's called gyrase. So helicase is going here, gyrase is going over here. Gyrase is also important to undo the winding, okay, to relieve the winding pressure. Okay, so let me see here. How is it that DNA gyrase is working? Okay, it's going to cut a little bit of the DNA and then unwind it and then seal it back together. Okay. Okay, so now we go into the bidirectionality here of your replication. All right, so here we're going to we open up the DNA. Now we're going to get to actually making the new DNA. DNA polymerases cannot start new synthesis of DNA. They just can't. Okay? So how they're going to do it? Well, they're going to rely on another protein that is going to start the synthesis. This protein is called the primase. It's going to make a little primer that is going to start the synthesis of DNA. But this primase can only start the synthesis by using RNA. Okay? So the primase is going to make a little strand of RNA, and then DNA polymerase is going to go there and continue the synthesis. All right? So primase is starting with RNA, DNA polymerase picks it up. Okay. For prokaryotes, the DNA polymerase that is responsible for most of the new strand synthesis is DNA polymerase 3. Okay? Now, polymerase 3 is very good for the leading strand, and basically the leading strand doesn't need anything else. For the lagging strand, you also have this other polymerase that jumps into action to remove the primers that are RNA and put in DNA. Okay, so DNA polymerase 1 is the one that goes at the Okazaki fragments and kicks out RNA and puts in DNA. Okay? And it does that like in one sweep, like kick out, put it in, kick it out, put it in. Is really quick and effective at removing those primers. Do you have other types of polymerases? Yeah, but you're not going to worry about them. You're going to worry about 3 and about 1. Again, so keep in mind here, your DNA polymerase cannot start a synthesis, but it can continue a synthesis because it needs that free 3' OH group. Okay, no OH group, no synthesis. Okay, so you got your primer. Okay, all right. So here is a bit on those scientists who actually figured this one out. And look at this. Oh, DNA polymerase 1 was discovered first, and then later on you had DNA polymerase 3. This is like 1970. This is not a long time ago. Okay? I was born in 1972. So that's scary. That's not very old at all. Yeah, it's not. So, so here, DNA polymerase 3, right? In 1970. And then so much happened afterwards, right? Okay, so what is happening here? You're going to have your synthesis 5' to 3'. What is the energy that is going to be used for the synthesis? It's going to be pyrophosphate. 2 of your phosphate groups get chopped off of the triphosphate. Okay, so here a little movie showing their pyrophosphate getting kicked out. There, and here you have your nucleotide that gets added and added, and you have your base pairing, right? 3 hydrogen bonds or 2 hydrogen bonds right there, and that's the job of polymerase. Bringing in nucleotides, cutting out the pyrophosphate, and adding that to the new strand of DNA. Okay. So here is actually how your DNA polymerase 3 looks like. So yeah, this alien looking thing, that's your DNA polymerase 3. Okay? It has 2 arms. It's tethered in the middle. Okay? So one arm is going to be doing the leading strand. The other arm is going to be doing the lagging strand. Okay. So different subunits, okay? What are the subunits of your DNA polymerase 3? This is what you gotta remember, okay? You have the alpha subunit. Where is alpha? Right there, right here. The alpha subunit right here, that is gonna make the polymerization of the DNA, okay? Alpha subunit right here. Then you have your epsilon proofreader. Epsilon proofreader is right here, is right close to the alpha subunit, because it has to make sure the alpha subunit did a good job. If alpha did not polymerize right, the epsilon subunit is going to make sure that this alpha goes back and redo its job. So our epsilon is your proofreader. Okay. Besides all that, you also have this stimulator up there. This theta stimulator. It's going to make sure everybody's working. Okay. Now, besides this polymerizing unit, you also have the eyes of this alien, and the eye is actually called a beta clamp. It's extremely important for lagging strand synthesis. Okay? Because this beta clamp has to make sure that polymerase 3 stays stuck on the DNA and does its job. Okay. Now, what does that mean? If you don't have a beta clamp, your alpha subunit gets there, and guess what happens? You see this alien guy over there? The alien-looking guy over here? Without those eyes, this alien has a very short attention span. Okay? What does that mean? It's like, oh, I'm going to polymerize DNA. I'm polymerizing DNA. Oh, butterfly. I fell off the DNA. Oh, I have to polymerize DNA. Oh, let me start again. So that's the problem of low processivity. We don't call it attention deficit. Okay? We call it low processivity. With the beta clamp, what happens? Beta clamp goes in there and make sure the alpha subunit stays associated with the template and doesn't fall off. Okay? So beta clamp goes in there and alpha can do the polymerization because beta clamp is actually doing a little levitation trick on the negative strand of DNA. Now look at the beta clamp. You see here this protein right here? Everybody sees the beta clamp right there? It is associated with your string of DNA. What charge is the DNA? Negative. How do you think this thing is associated with the DNA? Positive charge, right? This is a little levitation trick of your beta clamp. What do I mean? Here is a clamp. It has a lot of positive charges. These are positive charges. And then DNA goes right in the middle with a negative charge. So you have a little magnet going on over here that is levitating in this negatively charged string of DNA. Really cool. And it doesn't get the thing stuck because it's levitating. Okay? You have this charge interaction going on over there. I'm going to take 5 more minutes to talk about that. Okay. How does the clamp get there? You have this other subunit that is called the gamma loader. Means the gamma clamp loader. Okay. So look at this alien. Let's find the gamma right here. Okay. So this part here that looks a bit like the nose of the alien is the thing that is going to load the clamp to the DNA. How is it going to do it? It's going to do that magician trick with the ring and the string. Have you seen that one? The magician will let it up, and by magic, the string gets stuck to the clamp, to the circle. You know what I'm talking about? No, nobody went to circus around here? No? I mean, YouTube, magician trick. Okay. You have a clamp, you have a string, magic. So this clamp loader is the magician. He gets the clamp, opens up the clamp, put up the DNA, and that's the job that it does. Okay? Is the clamp always going to be associated to the DNA? No. Pay attention because everybody forgets this detail and it's important. The clamp is only associated to a single strand of DNA. It doesn't make sense for the clamp to be associated with double strand of DNA because that one is already polymerized. So when the clamp sees double strand, either DNA or DNA RNA hybrid, which is the case in the lagging strand, right? When the clamp sees 2 strands, it will fall off, okay? And this is when the gamma guy picks up the clamp and puts it back on the part of the DNA that is single strand for a new stretch of polymerization. Everybody got that? Clamp association? Okay. What is the importance of the clamp? Increasing the processivity. This is for bacteria. The clamp name for eukaryotes is called pCNA. Don't ask me why. But yeah, the beta clamp for eukaryotes is pCNA. We'll get there. All right, so clamp loader and the clamp. And here we go for the primers that start the synthesis, and then for elongation with your polymerase. Okay, so continuous leading strand, discontinuous Okazaki in your lagging strand. All right, so now, how do you get rid of the RNA pieces in the Okazaki fragments? This is when you get your polymerase 1. It goes there, kicks out the RNA, puts in the DNA, and then it's not going to kick out the DNA. When it doesn't see any more RNA, it falls off. But look at this. Polymerase 1 cannot do the final joining of this newly synthesized DNA strand over here because it doesn't have a leaving group to add any more nucleotides on the strand that is already there. So it doesn't do anything. It leaves a little empty space, a little nick over there, and it can't do anything else, so it falls off. The very, very important enzyme that goes in there and seals the nick is this guy over here, DNA ligase. In order to seal the nick, it needs energy. It needs ATP. So it goes there, uses ATP, makes a bit of an activation of the leaving group, makes the group leave, and it seals the nick. Ligase, very, very important enzyme. not only for regular DNA replication, but also for cloning. Okay? If you want to clone a piece of DNA, the only way you can do that is because we have ligase to do the job. So ligase is very, very important, and this is how ligase does the job. You have here a 5' phosphate. Remember, polymerization goes 5' to 3'. So here, you don't have the energy of the leaving group on that nucleotide. So ligase goes in there, and it uses ATP to artificially put there this bulky, energetic leaving group. Okay, so it activates the 5' end of your nick. Now you can have a nucleophilic attack and a leaving group that comes out in order for your backbone to be sealed together. So this is how ligase does the job at the Okazaki fragments. Okay, make sure you understand this, because am I going to ask you about this? Yes. Okay. If anybody gets this question wrong in the test, I'll be very, very upset. Okay, so let me see here what else is important. All right, proofreading. Why is it important to do proofreading? Because if you don't, then you get mutations. Mutations happen, okay, but you don't want them to happen very often. Otherwise, your cell can die. The bacteria is going to die, but it doesn't matter, they are replicate. But if your cells die, then that's a big problem, because they can, well, if they die, it's not a big problem. The problem is if they don't die, it becomes cancerous cells. Then you have a bigger problem. Okay, so let me see here. I'm going to stop at this slide, but pay attention, because I have, 12 slides that I'm going to add the video to the end of this video, and I'm going to hold you responsible. All right? Make sure you see the differences for your eukaryotic and prokaryotic. Basic stuff is done.